Hello, everyone, and welcome to the March meeting of the Global Health Youth Organization. My name is Nooraldeen, and I'm a co-founder of the organization, and we are very excited to see everyone who joined us today. We want to thank everyone for joining the meeting and working around any scheduling or time zone issues that you might have had. As for the format of today's meeting, we will begin with a quick introduction of our speaker, followed by her presentation, and then a question and answer session. After the Q&A section, we will post a shadowing certificate link in the chat. We ask that all members stay muted throughout the presentation so that we can limit interruptions and distractions. Please feel free to add any questions or comments that may arise in the Zoom chat. I will now be passing it over to Adam, the other co-founder of the organization, to introduce today's speaker.
Thanks, Nor. Hello, everyone, and as Nor mentioned, I'm a co-founder of the organization. It is so great to see all of you that joined, and we are so happy that we're able to attend this meeting. We want to once again thank Dr. Cheng for taking the time out of her day to present today. To quickly provide an introduction, Dr. Cheng is a postdoctoral research scientist at Boehringer Ingelheim Pharmaceuticals, and will be speaking about her research on oxidative stress and aging. Please remember to stay muted throughout the duration of the presentation to minimize distractions. Again, if any questions do arise, please type them in the chat and we will get to answering them at the end of the presentation. With that being said, we will now hand the floor over to Dr. Cheng for her presentation. Thank you.
Hi, everyone. Thank you, Adam, and thank you, Nooraldeen, for the introduction. And I really appreciate this opportunity to talk about my research and to share my research and my career experience with you. And today I will be talking about actually my PhD research on oxidative stress and aging, because I feel this is a pretty interesting topic, and I hope we can discuss more about the impact. And if you have any questions about me or about aging, oxidative stress, or about my career, please feel free to connect with me through my LinkedIn. And if you have any questions, please type in the chat box, and I'm happy to answer them. All right. So the topic of my research of my talk today is broccoli, oxidative stress, and aging. So I will focus on the last two keywords in the most part of my talk, and the broccoli story will come at the end. All right. So, oops. All right. Let me change to laser pointer. All right. So before I start, I just want to give you an introduction about myself. So I am a biologist. I get my bachelor degree in a very beautiful university in China called Xiamen University. Over there, I had a brief experience with liver cancer research, and particularly I was working on microregulation of liver tumor genesis. And after my bachelor, I came to United States, and I studied actually genetics in the University of Rochester in upstate New York. And I worked on oxidative stress, epigenetics, and the aging process. And of course, when you talk about Rochester, you talk about snows, right? So I have this very cute picture about University of Rochester in the snow. So it was a pretty impressive experience for me. And after that, I joined Boehringer Ingelheim, which is a pharmaceutical company at Ridgefield, Connecticut. And here I worked as a postdoc fellow, working on early drug discovery. So although it's a pharmaceutical company, I still work on very early upstream drug discovery research. So here I worked on basically identifying new potential drug targets to promote intestinal repair and to treat inflammatory bowel disease, which is a disease caused by chronic inflammation in our gut. All right, so here is a very nice picture of our Boehringer Ingelheim at Ridgefield. All right, so as I mentioned, today I will be talking about one of the projects that I worked on during my PhD research about oxidative stress and aging. Before I start, I just want to thank actually everyone who contributed to this project. My PhD advisor, Dr. Dirk Bowman, he is a very cool, very smart scientist, and I learned so much from him. And everyone in the lab who contributed to this project. It was a very pleasant 6 year of me doing my PhD research. All right, so I will get started. We all know that aging is the process of getting old. In fact, why we age, how to delay the aging process, whether we can like rejuvenate ourselves after when we are getting old. These are very, very interesting research topic that has been like attracted so many researchers. People through so many years are trying to find ways to delay aging, to rejuvenate ourselves, and so on. And there are so many theories about why we age, how we age. Some of them are like scientific theories, some of them are like philosophical theories. It is a very fascinating field to study. So one of the theory, or I shouldn't say the theory, one of the fundamental phenomenon during the aging process is that there is this accumulation of macromolecule damage in our body. So the damage to our DNA, to our genetic materials, to the proteins, all these contributes to the loss of physiological fitness during the aging process. And this is something that people are trying to avoid, or trying to delay or counteract, to find ways to delay the aging process itself. So we know that stress can induce damage in our body. And actually, our body during the whole like evolution process has evolved such good and efficient stress response systems to counteract or to fight with these internal or external stresses. So here, this is a very, very simple illustration of a stress response in our body. So when we feel a stress, or when we sense a stress signal, the body turns on the stress response system to counteract with such a stress-induced damage. And after the stress is solved, it came back to normal basal condition. And usually, as you can tell, this is a very dynamic, very robust system. And our body actually rely on this robust stress response and repair system to maintain a state we call homeostasis. And here I listed a few sort of well-known cellular stresses, including oxidative stress, or free radicals, or reactive oxygen species, ROS, or heat shock stresses, or unfolded or misfolded, like protein folding issues, or of course pathogen-induced, for example, inflammatory or immune related stresses. So all these stress need to be well addressed to maintain a functional physiological condition. And in fact, it has been reported in many studies that during the aging process, the robustness of many stress response systems declines with age. So instead of having this very crisp, very accurate response to specific stress signals, in many cases in the old animals, such response is lost. And as a result, you can tell these old animals or old organisms cannot really respond well to the stress or to the environmental internal challenges. And probably as a result, there are more damage, stress-induced damage in the old organisms. So, of course, a fundamental reason is what happened during the aging process, why the stress response system lost their responsiveness during the aging process. And more importantly, what we can do to inhibit or to delay this process, and to hopefully preserve a more healthy, more functional state in our body. All right, so today, the stress that I'm going to focus on is oxidative stress. So I believe you all hear about oxidative stress more or less. It plays a key role in many cellular processes, and it has been proposed that oxidative stress is a contributor to many diseases. Here, I just listed a few from this NIST image. For example, lung disease, heart disease, or neuronal like Parkinson or Alzheimer. These are all age-associated diseases, and gut diseases and cancer. And aging itself has been proposed that oxidative stress is a contributor, or oxidative stress induced damage is a contributor of aging itself. So I am pretty sure you are familiar with antioxidant supplements. There are so many antioxidant supplements in the market. However, in terms of treating diseases and also about treating aging, for example, lifespan extension, they are not working as successful as one may expect, because people has been proposed that oxidative stress plays such a key role. You would expect that the antioxidant may work very efficiently. However, it is not. So there are many research articles discussing about this one. There are issues with like how much you take it, or what the time, like the timing you take it, and how an antioxidant is metabolized in our body, and so on. And more importantly, people start to realize actually the reactive oxygen species, or just oxidative stress, is not all bad. So you do not want to kill all of them in your body. Sometimes they can be important signaling molecules. So therefore, you do not want to basically have a lot of antioxidant to neutralize everything. You somehow want to maintain it as a very healthy and organic level. And how do you do that? So the goal of our research, and many other research as well, is to, instead of taking antioxidant, we want to preserve the function of our intrinsic antioxidant defense system. And that came back to this model. We want to preserve the robustness and the accuracy of our intrinsic antioxidant defense system throughout life, to hopefully have a healthier way to fight with oxidative stress. All right, so how do we preserve the intrinsic antioxidant response? Here, I want to introduce you a key intrinsic antioxidant regulator, a gene called NRF2. And there are other regulators as well, but today's talk will be focusing on NRF2. So it is a gene that's expressed in most of our cells. And how does NRF2 work? So when a cell sees a challenge from an oxidative stress, it activates NRF2, this protein or this gene. And what NRF2 does is that it can activate the expression of many cellular antioxidant genes. Here I listed a few, which will be used later in the talk as a sort of a reporter, or a way to access NRF2 function. But basically, NRF2 turn on all these antioxidant genes, and this can again fight the oxidative damage. So the first question actually we asked is, how does NRF2 signaling change during the aging process? So this is supposed to be a healthy condition, but what happened in the old animals during the aging process? The same thing also happened. And before that, I just want to let you know that we use fruit flies as a model animal to study this. Well, fruit flies has been used in many biological research. They are actually one of the very famous animal models, and they have great genetic tools and all the fantastic tools. But one particular advantage of using fruit flies for aging studies is that they live a short life. So instead of working with mice, you have to wait for a year to wait for the mice to get old. In flies, you only need to wait for a couple of weeks. So it shortened the timeline of people's research. Therefore, it's actually is a very good tool to study the aging process. All right, so for some of you who may be interested in the molecular mechanism of NRF2, so here is it. So NRF2 is a category called transcription factor. When it's activated, it is transferred into the nucleus and bind to the bind to the genome. And here, after binding to this after binding site, it can turn on the downstream gene expression, and these genes including GCLC, those antioxidant genes. So here, this is another very important protein called MAF, as I shown here. So MAF, as you can tell, is a protein, and it served as the binding partner of NRF2. So MAF sits here all the time, and when NRF2 activates itself, it's activated by oxidative stress. It came in and bind to MAF, and when it bind to MAF, bind to the DNA, it activates the gene expression. So MAF actually is going to be the focus of the first part of my research. All right, so let's get started. So as you can imagine, our question is what happened to NRF2 signaling during the aging process? And the first experiment we did is to compare young and old animals and see what happened to their NRF2 signaling. Here we used a technology called reporter flies. So in these flies, the green color, which indicates the expression of a protein called GFP, or green fluorescent protein. So this protein expression is driven by NRF2 activation. In other words, when NRF2 is activated, the protein is expressed. So as you can see that in young flies, after oxidative stress, you see this nice induction of this protein, suggesting that the pathway is activated upon stress. This is what we predicted, right? However, in old flies, you do not see this anymore. So even after stress, you do not see this higher expression of the protein. It suggested that the pathway or the NRF2 signaling is no longer responsive to the stress. And we also confirmed it with endogenous gene expression. And again, while in the young flies, we saw the induction of this gene expression after stress. In the old flies, we do not see this. So suggesting that the signaling, NRF2 signaling, is decreased with age, or is no longer responsive after during the aging process. So we next found an interesting phenomenon. So if we provide extra MAF, remember that binding partner of NRF2, we can counteract this process. So here, if you first look at the reporter flies, again, the young flies, either the control flies or flies with extra MAF, after stress, you see the induction of the green, right? However, in the old control flies, as we mentioned in the early slide, there's no longer induction after stress. But in the old flies with extra MAF, you see this induction of the green color, suggesting that this old flies with extra MAF, they still preserve a very responsive NRF2 signaling or antioxidant response in these flies. And again, this is also true with endogenous gene expression as well. So not only at the gene expression level, but we also measured the overall fitness of these animals, either the control animal or the flies with extra MAF in the old flies. So here we have 3 measurements. We measure the stress tolerance. Basically, we give the flies a lot of stressor, and we measure how long they can survive under this condition. And the longer you can survive, meaning the more healthy you are, right? And the next experiment is the climbing ability. We measure how much the fly can climb in a certain time. And again, the higher you can the higher you can climb, meaning probably the muscle function is better, and in general, it's more fit. And we also measure the heart performance by measuring the arrhythmia index, and the higher of the index, meaning the lower the heart function. So here, I hope you can appreciate that the orange color indicates flies with more MAF. And in all three assessments, the orange color ones perform better than the gray ones, indicating that flies with old flies with extra MAF can preserve in general more fit in their old age compared to the wild type or the control flies. All right, so at this point, what we know is that as we mentioned in the earlier, in the introduction slides, the NRF2 dependent antioxidant response declines with age. And by providing flies with extra MAF, we can delay the aging process. All right, so here I just want to let you know that actually this is not something unique to the flies. So actually in other studies, people have found very similar situation in other animals, such as in worms, in mice, in rats, and in primates. So this is really a conserved phenotype that happened throughout the animal kingdom, and people are really interested in what's going on there, whether we can find ways to delay this change. And all right, so now we have two other questions, which is actually the start of my independent research. So they are actually connected with each other. So my two question is, so firstly, what happened? Why do old flies have this inefficient signal responsiveness? And my second question is, what is the function of MAF during this process? Why does it helpful if we have more? So the first experiment we did to answer those two questions is by asking a very simple question: whether the effect we saw with MAF overexpression or with this extra MAF, whether this is a long-term or a short-term effect. All right, so here we designed an experiment where we provide flies with extra MAF in different fashion. We call them either long-term MAF overexpression or three short-term MAF overexpression. So in the long-term MAF overexpression, we provided extra MAF to the flies throughout their adulthood. So after eclosion, we give them extra copy of MAF through fantastic fly genetics tools. And we also have three short-term ones where we only express this extra MAF at the late stage, the early stage, and the middle stage. And we want to know in all four groups which one can recapitulate the beneficial effect we saw in the early stage, in the early slides. And the answer is the second one, the long-term one. So here I'm showing you two representative images. One is the expression of an NRF2 target gene, and the second one is the stress tolerance assay. And in both cases, we found that this long-term MAF overexpression or chronic MAF overexpression can delay the aging process, and these flies seem to be more fit in the old age. However, in these three short-term, we call them week 1, week 3, and week 5 MAF overexpression groups, we didn't see any beneficial effect. So here it suggested that MAF if we give them extra MAF, it needs to be a long-term process to delay the aging process. And especially the 5 weeks one suggesting that we cannot somehow reverse the process if we only give them more MAF when they're already old. In other words, we cannot really rejuvenating the flies. We can only delay the aging process. So yes, so this is the conclusion here. The long term is helping, but the short term is not. So then when we came back to our question, what is the function of MAF? At this point, we have two clues. One is that MAF need to be present all the time to achieve this beneficial effect. And the second one is, as I mentioned earlier, MAF is a DNA binding protein. It helps NRF2 bind and function as the transcription factor. So long story short, here I skipped some molecular like epigenetic stuff. But long story short, we came up with this hypothesis that MAF may work as a chromatin regulator. So if you imagine, we think that MAF serves as this placeholder to hold the chromatin structure so that NRF2 can still bind to the DNA and regulate downstream gene expression. And without MAF, this binding site, NRF2 binding site, may be disrupted so that NRF2 can no longer regulate its gene expression. And these are nucleosomes, which are basically chromatin structures. All right. So here I'm showing you one experiment we did to confirm this idea. So this is an experiment where we directly measure the chromatin, openness at this binding site. So this is a little bit complicated graph, but I want you to follow my laser pointer. So here, firstly, I want you to look at group 1 and group 4. So you can tell that in both two NRF2 target genes, in the young, if you compare the young control animal and the old control animal, there is a decrease of chromatin openness in both NRF2 binding site, suggesting that the chromatin or the DNA or the NRF2 binding site is no longer accessible in the old animals. And next, if you look at group 4 and group 5, so both in old flies, either the old control or old flies with extra MAF, you can see that MAF is able to at least partially maintain the openness of the chromatin structure. So here we think that we came up with this model that somehow during the aging process, the NRF2 binding site, which is somewhere here, which is supposed to be open, is no longer open, became closed or disorganized. And as a result, well, in the young flies, everything came function very well. So when NRF2 is activated, it binds here and activates genes. In the old flies, this place is no longer accessible. So MAF and NRF2 cannot bind to it even after stress. So the downstream signal cannot be activated by the stress. And somehow by providing the flies with extra MAF throughout the aging process, we can counteract this age-associated degeneration at this process, at this binding site. And here we think MAF work as a placeholder to hold this region open. All right, of course, there are more molecular magnetic study, which I skipped today just for the time's sake. But this is our current conclusion with the MAF story. And next, of course, everything I showed you earlier is achieved by overexpressing a gene through genetic ways. This is not very practical if we want to apply our findings in like a more practical condition. Therefore, we were looking for ways, like pharmacological ways, whether we can, for example, feed the fly something to have similar effect, like small molecule or pharmacological agent. So we came up with this idea based on this very old saying, use it or lose it. We want to see if we keep using the pathway, whether we can maintain the function of the pathway. It's like exercising the pathway. It's like how we exercise our muscles, right? We keep using it, so we exercise it. So we want to see basically if we can preserve the NRF2 function by continuously activating the pathway. So here we utilized a NRF2 activating molecule called Oxidative. And of course, now there are so many molecules that can activate NRF2 through direct or indirect ways. And here, because our lab has a history with this molecule called Oxidative, so we just use it. And I should mention at the beginning that this is just purely exploratory. There's no clinical, nothing related, nothing confirmed, and everything was done with flies. So it's very early stage. All right, so as I mentioned, we want to see if we keep activating the pathway through this activating molecule, whether we can preserve NRF2 function. So we basically designed our experiment based on this idea. So we have, in this experiment, 5 groups. We have flies under control conditions, so no alteplase was added to these flies or treated to these flies. And then we have 2 long-term alteplase treatment group. We call them continuous treatment and intermittent treatment. So continuous is simple. Basically, we feed the flies with alteplase every day throughout 5 weeks until they get old. And the intermittent, actually, we feed the flies with alteplase 3 days per week, and the other 4 days it's off alteplase. And then we also have 2 short-term groups. We call them week 1 group and week 5 group. So early stage and late stage. And want to see that at the end, which group can show a healthier condition, basically. And the first experiment we did is with reporter flies. Here, just want to show you a nice picture of these flies. I don't know whether you think it's nice or not, but I think they are nice. But we here first did the experiment with the reporter flies, again, the green fluorescent flies. And you can see that the continuous group and the intermittent group after stress, here we use a stressor called DEM, after stress can still induce the GFP expression, right? However, in other groups, the expression is no longer there. So it suggested that the long-term alteplase treatment can counteract this loss of signal responsiveness. And we can achieve this by either chronic treatment or intermittent treatment, but both has to be long-term. And this is also true with endogenous NRF2 target gene expression and with the stress tolerance assay. In both cases, again, we saw a beneficial effect when we feed the flies with the either continuous or intermittent alteplase strategy. And the short term is not working, just like math. And next, we ask whether this also related to the openness of the chromatin structure. So we measure the chromatin openness in these old flies. And again, we found that the continuous and the intermittent treatment were able to promote a more open chromatin structure in the old flies. And actually, if you can tell, intermittent actually works better than continuous. This is also very encouraging because if you think about using this, intermittent is definitely more practical than continuous, right? And this is a non-targeting gene, so there's no change. These 2 are 2 target genes, so there are changes. All right, so lastly, we asked whether alteplase treatment can promote lifespan extension in these flies. So here we tested control flies, continuous flies, and intermittent flies. So we tested in both males and female flies. And in males, we found really encouraging result. We found that both intermittent and continuous are able to promote a lifespan extension in these flies. And actually intermittent work a little bit better than the continuous, which is consistent with the openness of the chromatin structure. In the females, it's less encouraging. It seems actually not seems in terms of statistical analysis, intermittent is able to extend, although not too much, the lifespan. Continuous are not. So suggesting that it seems that alteplase treatment is able to promote a lifespan extension, at least in the males. But if you look at all the other, like the gene expression and everything, it's definitely has a higher or more impact comparing to the lifespan. So here, the many other studies has found similar things. They found that it seems that when they give the animal some treatment, the animals seem to be healthier. But when you measure the lifespan, there's not too much increase. Therefore, now the field came up with this health span versus lifespan. So sometimes the health span and the lifespan may not be directly correlated with each other. In other words, some animals may live exactly the same but healthier than the other animals. This is also something that we want to investigate in our future research in this project. And also the difference between male and female is also interesting. And but this is very typical when you do aging related study. It seems that male and female has different regulatory networks in their body to in terms of just longevity. All right, so here to summarize everything today, I talked today, we found that the NRF2 dependent cellular antioxidant response, this is the intrinsic response, declines with age. And one of the reasons for this is the disorganization at the chromatin structure. So it became more closed up. And this caused a loss of gene inducibility. So antioxidant genes cannot be induced upon stress in the older animals. And this result in an inefficient stress response in the older animals. And by providing flies with extra MAF or by activating NRF2 signaling through NRF2 activators such as Altifras, both in a long-term fashion, we can delay the aging process. But we cannot reverse the aging process when the flies are already old. So in this particular project, I did not find ways to rejuvenate the old flies. All right. So one of the future perspectives for this project, which is something that I'm really interested in, is whether the same concept can be translated to other stress responses, and such as like immune response or unfolded protein response, autophagy related, and everything else. And if so, whether this use it or lose it concept can be used to also preserve the function of other stress response. For example, whether we can preserve a efficient immune stress response by frequently get challenged by some at a healthy level by some pathogen. Of course, this is very naive idea, but this is something that we are trying to look into at this point. All right, so the last slide is about broccoli. So I talk a lot about Altifras. But Altifras has been used in clinical trials to treat, I think, in cancer-related clinical trials, but it didn't go anywhere because of the lack of efficacy and also because of the toxicity of the molecule. So, however, if you want to really boost your NRF2, there are molecules in the vegetables that we eat every day that can also activate your NRF2 signaling. And the vegetables such as broccoli, cabbage, kale, or cauliflower, they contains this NRF2 activating molecule that may be helpful for you. And also, many study has shown that physical exercise were able to generate a healthy level of stress for you, which is good for you. So the take-home message may be cliche, but eat the vegetable and do your exercise, right? So that's all the talk for today, and happy to take any question. Thanks.
Thank you so much. That was a really insightful and amazing presentation. So we will now begin the Q&A session. We will first start with the questions that were asked through the registration form, and then we'll get to any questions that were asked in the Zoom chat. So this is your opportunity to ask questions. So ask whatever you have in your mind. So to begin the Q&A section, the first question that was asked was how many and which pharmaceuticals have you worked on?
Yes, so I didn't talk much about my current research. So I am a postdoc research at Boehringer Ingelheim. I work on very early drug discovery research. So I actually technically I haven't worked on any pharmaceuticals directly. My current research is more related to drug concept development. So still pretty basic biology, and we're looking for potential new drug target, our genes or proteins that we can target to treat diseases. So it's kind of, I should say, at least at this point, my current research is kind of similar to the research I have in the academic labs. But that being said, we do have opportunities if one drug target turn out to be a very interesting target. We'll be able to involve more downstream development with like pharmaceuticals development or chemist or antibody generation biologist to work more on that. But personally, I do not have any experience with that.
Okay, thank you for the answer. And the next
Mhm.
question is there any clinical or patient care work in pharmacology?
So yes, there are. I believe again, I'm at this very upstream of pharmaceutical or drug discovery process, and the patient care, I guess, is the most downstream part. So I'm very far away from it. But actually, we all know that everything we do is for the patient. So actually, this is one of the most important topic I believe, in every pharmaceutical company, in the whole pharmaceutical industry. And for us, we actually have we invited patient to come to our company and give a talk almost routinely. And it depends on like what diseases we're working on. And over there, we were able to directly communicate with them. And so I think the company, all the pharmaceutical industry, they are very one of the very important part is to build a connection with the patient. So there are definitely patient care related work there. In terms of what specific ones, I'm not very familiar. Yeah.
Thank you so much for the answer. So the next question is, what interested you in a career in scientific research?
Mm, how do I say everything? Yeah, I started my lab work in my undergrads, and then I did a about 6 year PhD, and now it's my third year postdoc. So I like the process, and I just like the process of exploration. I just feel I like independent research. I feel more free. I was able to ask a question and find answers myself, and get challenged by others or by myself or by like the negative result of an experiment, and find ways to troubleshoot, solve the problem. I feel this is a very exciting and very rewarding process, and this is actually why I like scientific research. Yeah.
Thank you. That was very well explained. And the next question is a three-part question. How do you manage a schedule in the medical field? Is it difficult to manage your time? And do you have any tips for how to manage your time better?
Yes. So yes, for time managing, firstly, I'm not technically, I don't think I'm in the medical field. I'm not a doctor. Um, but still, I mean, I guess it's very typical that because now people are so busy and it's difficult to manage the time. The tip that it worked for me is to think about prioritization all the time. So it's not important. It doesn't mean that you have to do the right thing all the time or anything. I feel it's more important to have the habit to think or to prioritize, to train your habit to consider prioritization every day. And this is something that can help me manage my time and also reduce my stress when if there are a lot of things coming together, I may be stressed out and I may worry too much. But then if you think over and make your prioritization list, what you to do, one or two or three, I think it definitely helps. So yeah, that's something that I have been trying to learn myself as well. But hopefully I can share this with you. Yeah.
Yeah, that's a super great tip. Just prioritizing. That's really great. So How has the pandemic affected your career, and how have you adapted to continue to work and provide for others?
Yes. So I think I mentioned to you, Adam and Nora, at the beginning. I think the pandemic. One thing that's good for the pandemic is that actually we were able to connect with so many other people so far away through virtual, like Zoom, right? That's something that I think it's a good part of pandemic. The other thing for me personally, I work in the lab. I'm a lab scientist, and I have been so busy since I joined BI doing all the research, all the lab work. And then during the pandemic, at the early stage, my lab work was paused for about 3 months due to the whole social distancing consideration. And at that time, although my experiment, some of my experiment was delayed, I actually got more time to think over my project because I cannot do it. So I have to come home and think more and maybe prioritize more and read more papers. So I think it gave me a chance to rethink my project and to reconsider some work I did and some plans I'm going to do. So of course the delaying of my project was not very good, but I think it helped me to reconsider. And then this is a good opportunity for me because it barely happens before the pandemic. Yeah.
Thank you for that answer. And the next question is, what are some of the major advancements in the pharmaceutical industry?
That's a very big topic, I think. Um, so I think most of the biggest advancement came from the advancement from the science and technology. So it's not like because science, I believe I'm a scientist, and I believe science is the base, the foundation of the pharmaceutical industry, science and technology. And if you think about it, maybe I guess in the 80s, all the drug came from the chemistry play a key role in drug industry. They isolate compounds from animals from plants, and then they were developing ways to treat people. And there is the breakthrough came during early 2000 when the Human Genome Project came up, and people were able to sequence our genes, right? And they were able to compare a healthy person and I guess a cancer patient, and they were able to find, for example, cancer-related genes, and then they were able to target those, to design targets of those genes. So that advancement came from the advancement of the Human Genome Project, right? Now everything starts to be at single cell level, because when people are looking at genes because the advancement of single cell technology, now people have a higher resolution to look at not only what happened in your liver, but what happened to all the individual cells in your liver, and find out which cell is pathogenic to this disease. So that also thanks to the advancement of all the single cell based platforms, right? And there are other like the CRISPR. I'm not sure whether you know this, but you now have a much easier way to edit your gene using this CRISPR technology, and also the stem cell. So the Yamanaka won the Nobel Prize in, I guess, 2014, right? So he developed ways to de-differentiate your cells from a differentiate cells to iPS cells, induced pluripotent stem cells. So in this way, it allows you to, for example, if something happened to my liver, I can get some cells from my skin and redifferentiate to my liver cells and put it back to my liver. Of course, this is very naive, but that's also, this advancement also came from the whole area of stem cell technology. So again, sorry, maybe I talk too much, but I think all the the advancement in the pharmaceutical industry came from the advancement of the science and the technology.
Thank you so much. That was a super broad question. You gave such a detailed answer. So thank you so much.
Thank you.
So for the last question here that was asked through the registration form. So what is some advice you can provide for someone wanting to pursue a career in postdoctoral research? And what would you recommend to prepare yourself for?
Okay, so first of all, a postdoc is not a career. It is a training process. So you do not want to pursue a career in postdoc. Definitely do not, because so it's supposed to. So usually, who so usually people after they get their PhD, um, if they still want to stay in the research field, sometimes it's better to do a postdoc because it's a time for you. It's like a bridge from being a PhD student to being like a full independent researcher, like a professor. And postdoc is a nice bridge. And whether you need this bridge or not, if you're smart enough, you can after a PhD, you can directly be turned into an independent researcher. Or if you want to do like a short postdoc or long postdoc, it depends on you. But it is not it's supposed to be a transient retraining process. So it's not a career. And whether you want to do a postdoc or not, so I think that before you consider postdoc, just ask yourself, do you like research? Do you like what you want to do in the future, and whether a postdoc is necessary? So for example, if you get your PhD and you want to be a professor, you want to open your own lab, being a principal investigator and having your own PhD and postdoc students, then I think a postdoc training will definitely be helpful. Over there, you can learn more about not only about science, but also about managing a lab, about writing research proposals, writing grant proposals, and collaborating with other labs. All this, I think postdoc is helpful. If you get your PhD and you want to join industry, like being an industry research scientist, then the postdoc may not be necessary. It depends on your situation, because some industry is more looking into the skill set. If your skill set fits in their need, you may not even need a postdoc training. Of course, a postdoc would also be very helpful in that case as well. And if you get your PhD, for example, in genetics, and then you want to be a genetic consultant, then you don't need a postdoc at all. It's not necessary. Or if you get your PhD here in biology, and then you want to be like a patient care worker or a marketing person or whatever, then postdoc training is definitely not helpful. So it depends on what you need in the future. Just make sure you think over that before you start any. Yeah.
Thank you. That was very well explained. So now we're going to go over to the Zoom questions. We only have one question because the other questions were repeats that you already touched on. So,
Okay.
So for the question I was asked, so what do you believe the primary application of your research could be?
So my current this research that I talk about today, I think it's more basic biology. Although we touched upon the pharmacological concept, it's very, very early stage. So I think the primary application is for us to understand to have a more understand on the aging process and on the molecular mechanisms that contributes to this loss of stress responsiveness in the aging process. And because the whole aging research is still I should say at early stage, I don't think there are any current application of or drugs that could delay the aging process. So it's very early stage. So I think the primary application of this research that I talked today is definitely to have a more basic understanding of NRF2 signaling, of aging process, and of the oxidative damage during the aging process.
Thank you for all those amazing answers. We'd
Sure.
once again like to thank researcher Cheng for taking her time today to give us an introduction to some of the most advanced research in oxidative stress and aging. Thank you all for attending today's presentation. and please use the link posted in chat right now to obtain your shadowing certificate. Thank you all so much for attending this meeting, and we look forward to seeing you in our next monthly meeting, and we will stick around for a couple of minutes to make sure everyone has access to the link.
All right. Thank you, everyone.